I'm Alexander Fedintsev. I'm from Russia, but now I live in Finland. I was working in the field of agent research, but now I am moved to machine learning. But still, agent research is my passion, and I'm doing theoretical work on my own. Many scientists trying to find the cause of aging inside the cell. They look for DNA damage, mitochondrial dysfunction, accumulation of protein aggregates inside the cell. But what if the true cause of aging is not in the cell, but out of the cell? So our cells live in an environment called extracellular matrix, and it is comprised of very long-living proteins with very low turnover rate. And these proteins naturally accumulate damage, different types of damage. It is glycation, carbonylation, and brand new type of damage called carbamylation, non-enzymatic, different types of non-enzymatic modifications. And I argue that they contribute to almost all hallmarks of aging. One of the most famous hallmark of aging is cellular senescence, and it is believed that it is due to DNA damage and loss of telomeres, and it is kind of protects cells from being cancerous. But it doesn't seem logical, because if it is a protection from cancer, why do they secrete so-called SASP, senescent associated secretory phenotype, which is comprised of pro-inflammatory cytokines, metalloproteinases, which are helping cancer to grow and to migrate, to metastasize. I think that, and new researches show that actually senescent cells are a very important part of wound healing. And I think the key is here. Senescent cells are playing a big role in wound healing. And this allows us to unite wound healing and aging via matrix, and I'll explain how it is. So, when we are wounded, our fibroblasts they differentiate into myofibroblasts. This is a type of cells that can rapidly secrete like collagen and other types of extracellular matrix proteins. But what if they start to proliferate and secrete these collagen molecules to patch the wound? But what if the amount of collagen will be too high? Yeah? It will result in fibrosis. It's not good for a living organism, and that's why our organism should know how to avoid it. And senescent cells are the way to overcome this issue. When the excessive amount of collagen is deposited, it probably increases stiffness of surrounding environment. And cells must sense it through mechanosensing receptors and that kind of stuff. And then they become senescent, probably via CCN1 protein, like signaling molecule, that induces a cascade, a genomic program to become a senescent. Why we need this cellular senescence? First of all, the first hallmark of cellular senescence is that a cell ceases to divide, stops dividing. It of course reduces amount of cells that produce collagen, which is positively which limits the fibrosis. And second thing, that they start to secrete matrix metalloproteinases, which degrade the excessive amounts of collagen. And then they secrete pro-inflammatory molecules which attracts immune cells that clears them, and so the wound is healed and no excessive collagen deposited. But with aging, we get these crosslinks of ECM, of matrix, which mimics the stiffness of this excessive collagen deposited. And this like leads to accumulation of senescent cells even without a wound, and they start to do their like job, but without a wound, and it is detrimental to our health. They secrete this inflammatory cytokines, metalloproteinases, which degrades the healthy tissue, and this is the working hypothesis. Why do we accumulate senescent cells? So another hallmark of aging is genetic instability, genomic instability. One of the simplest cause of genomic instability is the following. When cells migrate, they often go through micropores of matrix, and the cytoplasmic part is not stiff. It easily goes through these pores, but the nucleus of the cell is quite stiff, and when migrating through the pore, it often breaks. It is so-called nuclear envelope rupture, and this leads to DNA damage. And the most migrating cells in our bodies are immune cells, and so accumulating of these crosslinks, it leads to formation of these small pores, and it is detrimental for our immune system in direct mechanical way. And this is only two pieces of a puzzle. I'm working on more, and I think that 90% of hallmarks of aging could be explained by extracellular matrix or damage to other long-living macromolecules that are not inside the cell but outside the cell also. So this is my hypothesis theory, and I think that paying attention to consequences of aging, like eliminating senescent cells, will not allow us to live really long. So it's most like eliminating of senescent cells is more likely about health span, not for radical extension of our lives, and even for about rejuvenation. Yeah, rejuvenation is making our matrix soft, like it is in a baby.
This relates, I guess, in principle to what we degrades. One of the seven deadly things.
Yes.
And that was glycogen.
Yes, glycosens. Yeah, but it is underestimated the influence of this extracellular damage, accumulation of the stiffness of the crosslinks, different types of crosslinks, other type of modifications, because carbamylation is not about crosslinks, but it somehow also damages the protein. So we need to pay more attention to that part, because it's very important, it's crucial. Without this, we will not reach even 120, 30 years expected lifespan. So
how long do you think it will take to actually solve this problem?
It is a very, very complicated problem, and that's why I'm here to bring attention to this problem. It maybe will take several decades to identify all these crosslinks and create breakers for them. But we should start doing it right now. And now I know only one scientist working on that problem, David Spiegel, but I haven't heard any news from him for quite a long time. So we need to start working on that problem right now. It is very, very complicated, and without like putting efforts right now to that problem, we will not solve it in the nearest future.
Yeah, well, several decades is 120 years. So hopefully we can solve it before then.
Yes, hopefully. But we should start as soon as possible. Yeah.
Any questions? Where's Dennis now?
One aspect also of damage to long-living macromolecules is that we have one source of this, or not source, but one thing that is damaged inside the cell, but not in every cells, only in so-called post-mitotic cells, like neurons, like muscle cells. In these cells, the nucleus remain for the whole lifespan of the cell because it doesn't divide. And there are nuclear pore complexes. And nuclear pore complexes, they disassemble during the cell division and reassemble again, but in neurons they do not, because the nucleus doesn't disassemble. And they are susceptible to all kind of these non-enzymatic modifications, and hence it leads with time to leakage of the nucleus. So the proteins or other molecules from the cytoplasm can easily enter the nucleus of the neuron, and it is detrimental for the function of a neuron, and it might be the cause of such diseases like neurodegenerative disease, like Alzheimer's disease. It is commonly thought that Alzheimer's disease is linked to beta amyloid or tau protein accumulation, but recent failures of big pharma to treat this disease by targeting beta amyloid probably tells us that it might not be the cause. So maybe we should pay attention to nuclear pore complexes then, rather than targeting obvious things. So there are plenty of possibilities, and they are under-researched. No one pays attention to that, and it's I think it's the cause of our that lifespan extension trends are slowing down right now, and it might be the cause that we reach the bottleneck, and I argue that this is the bottleneck. So.